my name is Katya Brossart. I'm a medical science liaison with Natera Organ Health. I'm pleased to be presenting this article entitled Prenatal Diagnosis and Outcome of Fetal Hyperechogenic Kidneys in the Era of Antenatal Next-Generation Sequencing. The authors are Deng et al. From Shanghai, and this article was published in Clinica Chimica Acta, the International Journal of Clinical Chemistry and Diagnostic Laboratory Medicine, in January 2022. To provide a bit of background for this study, congenital anomalies of the kidney and urinary tract or CACT are the single most common group of anomalies prenatally diagnosed, and isolated hyperechogenic kidneys, or HEK, is something that might be seen on routine prenatal ultrasound or identified during scans for other indications. Causes of HEK range quite a bit from normal variation to underlying renal anomalies such as dysplasia, fibrosis, interstitial infiltration, glomerular or tubular dilation, or microcysts, and the range of etiologies or prognoses associated is even broader, and this really can complicate perinatal counseling and decision-making. Prenatal genetic testing, when available, tends to be limited to aneuploidy testing and testing for copy number variants, and the authors sought to better understand the full range of underlying genetic etiologies seen with HEK and to quantify this in a small cohort. This study involved 28 cases of fetal HEK diagnosed at a single referral center between 2016 and 2020. In this clinic, it was routine for this indication to offer genetic testing for aneuploidy and copy number variants. In addition, for the purpose of this study, when those results were non-diagnostic, trio whole exome sequencing was offered. Additionally, the authors performed a systematic review of the literature pertaining to genetic findings associated with prenatal HEK. The authors opted to group their 28 cases of HEK as well as the findings from their literature review into three broad categories. So one involved isolated cases of HEK, the second were non-isolated cases with normal amniotic fluid volume, and the third were non-isolated cases with abnormal amniotic fluid volume. So for the isolated group, there were 8 cases out of the 28. There were 16 cases from the non-isolated group with normal amniotic fluid volume. These involved a range of associated findings from polydactyly to additional kidney anomalies and a range of other anomalies associated with different organ systems. For the non-isolated cases with abnormal amniotic fluid volume, there were 4 cases, 2 involving polyhydramnios and 2 involving oligohydramnios. Looking at the genetic testing results, there were 2 cases, or about 7%, with aneuploidy. There was 1 case of trisomy 13 and 1 of mosaic trisomy 9, and 7 cases, or 25%, with copy number variants. 6 of these were those involving a 17q12 deletion, that is a recurrent deletion encompassing the HNF1B gene, which is commonly implicated in pediatric kidney disease, and then there was additionally 1 CNV that was a variant of uncertain significance. There were also 10 cases, or nearly 36%, with a pathogenic or likely pathogenic single nucleotide variant in a gene associated with kidney disease. So this is particularly striking because this was really what the authors were seeking to better understand and quantify in this group. And really, this is a group that is a group of etiologies that is not commonly tested for in the prenatal setting in this context. And so the genes implicated here included PKD1 and PKHD1 associated with autosomal dominant and autosomal recessive polycystic kidney disease, genes associated with Bardet-Biedl syndrome, and the HNF1B gene that I mentioned, as well as ETFA. There were also 3 cases, or 14%, with variants of uncertain significance that may be related to the phenotype. And then there were 10 cases in which there was not a diagnostic finding on the genetic testing. thinking about the implications of this study, I'd say the most striking finding is that in this small cohort of prenatally diagnosed HEK, the monogenic causes were so common. So the monogenic causes were the most common group of genetic findings in this cohort. The HNF1B variants were very common, and extending even out from the monogenic causes to findings of microarray, the 17q12 deletion syndrome was such a common finding in this cohort. Interestingly, there were cases of prenatal HEK in this group that had a genetic etiology that didn't necessarily lead to clinically significant disease in infancy, and this suggests that there are cases in which prenatal HEK may be kind of the tip of the iceberg, and that testing then might identify an underlying genetic etiology that really wouldn't otherwise become apparent until much later on. Particularly for isolated HEK, genetic testing beyond karyotype and chromosomal microarray analysis is fairly unusual in the United States, at least, and this is particularly interesting because this study suggests that monogenic disease, which is not so commonly tested for, is likely the most common cause of disease in this cohort. So obviously more work to be done. This is a small cohort, but very interesting findings. And I suggest that if you have time, you may want to explore this paper in some more detail. There is interesting discussion of the literature review as well as a deeper dive into the specifics of the cases that they were analyzing. Thank you so much.